A very good day to Dr. Leslie. Today, my group, Group 1, Subgroup 9, will be presenting to you about food contamination by Listeria monocytogenes in ready-to-eat food. My team members consist of 4 people: Nurul Alia Maisara, me myself Precious Luna, Pauline Kong Hui En, and Pamela. Listeria monocytogene is a gram-positive bacterium responsible for listeriosis, a serious foodborne illness. It poses a major risk in ready-to-eat food because it can survive and grow at refrigeration temperature as low as 1 degree Celsius without altering the food smell, taste, or appearance. This makes contamination difficult to detect and control. In ready-to-eat food, contamination typically occurs after the cooking process, during handling, slicing, or packaging, especially if hygiene practices are poor or equipment is contaminated. Since these foods are not heated before consumption, any bacteria present can directly pose a health risk. Symptoms of listeriosis include fever, chills, nausea, and muscle aches, and in severe cases, it can lead to meningitis. High-risk groups include pregnant women, the elderly, and immunocompromised individuals. In pregnancy, the infection can cause miscarriage, stillbirth, or newborn infection. Due to its resilience and ability to form biofilms, listeria is difficult to eliminate. Advanced input food biotechnology such as rapid detection tools and improved preservation techniques are crucial to ensuring the safety of ready-to-eat food products and protecting public health. There are several types of ready-to-eat food. So the first one is deli meats and cold cuts, such as ham and turkey slices. Secondly is the soft cheese, such as brie or products which are made from unpasteurized milk. And then lastly is the smoked seafood, such as salmon and trout.
The primary cause of Listeria monocytogenes contamination in ready-to-eat foods is its widespread presence in the environment. It naturally inhabits soil, water, and decaying vegetation, which leads to frequent introduction into food processing environments through raw materials and water sources. Cross-contamination during processing, packaging, or handling is a major route by which Listeria reaches ready-to-eat foods, particularly when hygiene practices are insufficient or equipment sanitation is inadequate. A critical factor contamination risk is Listeria monocytogenes' ability to grow at refrigeration temperature. Unlike many other pathogens, it can multiply during cold storage, which is common for ready-to-eat products with extended shelf lives such as deli meats, soft cheese, and smoked fish. Additionally, since ready-to-eat foods are consumed without further cooking, any contamination present at the point of sale or consumption can directly cause illness. Controlling Listeria in ready-to-eat food is challenging due to its resilience and adaptability. Recent research highlights its ability to survive under various stress conditions, including low temperatures, acidic pH, and high salt concentration commonly used in food preservation. This adaptability makes traditional preservation methods less effective. Another significant challenge is Listeria capacity to form biofilms on food contact surfaces and equipment. Biofilms protect the bacteria from cleaning agents and disinfectants, allowing persistent contamination source in food processing environments. Detection and monitoring remain difficult because Listeria can exist in low numbers that are hard to detect but still pose a risk, especially to vulnerable populations such as pregnant women, the elderly, and immunocompromised individuals.
A biotechnology solution for controlling Listeria monocytogenes in ready-to-eat foods, which is bacteriophage treatment. Bacteriophage are natural viruses that specifically infect and kill Listeria. They are approved by food safety authorities like FDA. Their mechanism is simple, whereby phage attached to Listeria cells inject their genetics material and cause the bacterial cell to reproduce, effectively eliminating the pathogen without affecting the food. This method is widely used in ready-to-eat food like smoked fish, deli meats, and soft cheeses. A common product is phage guard latex, applied by spraying and dipping during process. For example, a case study in Norway and Europe, whereby phage guard latex is used on smoked salmon and has shown over 99% reduction of Listeria without changing the product's taste and texture. This method is safe, effective, and increasingly used to improve food safety.
Moving on to the evaluation of the proposed biotechnology solution, where we'll discuss on the advantages and disadvantages of the solution. Let's talk about the advantages first. Firstly, it has high specificity, meaning to say that bacteriophages target only Listeria monocytogenes, leaving beneficial microbiota and food properties unaffected. Secondly, it has GRAS status and regulatory approval. Products like Listex P100 have been granted GRAS, or generally recognized as safe, status by the FDA, facilitating their use in food applications. Thirdly, they are effective against biofilms. Phages can penetrate and disrupt biofilms, which are often resistant to conventional sanitizers. Now let's move on to the limitation. Firstly, it has narrow host range. Phages are highly specific, which means a single phage may not target all strands of Listeria monocytogenes. This may require phage cocktails for broader coverage. Secondly, it has the potential for bacterial resistance. Listeria can develop resistance to specific phages over time, potentially reducing long-term effectiveness. And lastly, stability and storage issues. Phages may be sensitive to environmental conditions such as temperature and pH, affecting their stability during storage and application.
Listeria monocytogenes remains a serious threat to ready-to-eat foods due to its persistence and ability to grow in cold conditions. Bacteriophage treatments offer a highly effective and sustainable biotechnology solution, specifically targeting Listeria without harming beneficial microbes or food quality. However, ongoing research and updated regulation are essential to ensure safe, effective, and widespread use of this promising technology in the food industry.
That is all from our group, and thank you for listening.